Good afternoon, everyone. Thank you very much for coming. This is our last excellent series for 2023, so thank you for joining us this afternoon. My name is Katie, and I'm the Marketing and Events Manager for B-Link Innovation. I'm just going to run through the housekeeping really quickly before I hand it to the Vice Chancellor. As we start. So emergency exits. We don't have any fire drills planned, so if you do hear the alarm, it is a real evacuation, and the exits are just behind you, out those two doors, and the congregation point on the patch of grass. The toilets are just outside of these doors on the other side of the corridor. And after the speaking part has finished for today, we will be serving some food and drinks through in the cafe area. So please drink responsibly and enjoy whilst you network. That's it, keep it brief. So I'm going to introduce Lincoln University's Vice Chancellor, Grant Edwards, and he's going to introduce our speaker for this afternoon.
Tēnā koutou katoa. Welcome here today. It's great to see a really large audience for the last excellent series of the year. It is through our excellent series that the university is privileged to be able to bring excellent speakers to the table and contribute and engage in public debate on important issues facing this country and our communities and globally as well. It has been part of our professorial address series that is taking place this year. So this does actually equate to Eirian's professorial address, albeit a little bit late, having been promoted to a professor, I think, in 2022. This afternoon, we are going to hear from Professor Eirian Jones. Eirian is an applied plant pathologist whose research and teaching focus on informing the development of sustainable control strategies to provide solutions in the agriculture and horticultural industries. Her main research area is the development of sustainable control strategies for plant diseases, with a particular focus on biological control of plant pathogens and the role of indigenous microorganisms in disease suppression and plant growth. Her research has provided key information to the industries, a range of industries, including those of kiwi fruit, grapes in particular, and as key parts of her research. This afternoon, Eirian will discuss the outcomes of her 20 years research, aiming to unravel the complex interactions concerning the epidemiology and control of soilborne and woody trunk diseases, mainly focusing on grapevine trunk disease. She will discuss her recent work on the progress of identifying and engineering microbiomes for improving plant resistance to these intractable diseases. We welcome Eirian here today. Thank you very much.
Thank you for that introduction. So, as Grant said, what I want to talk about a bit today is the work that we've been doing here at Lincoln with my group of students that are all over there on trying to understand pathogen systems, so trying to understand how these pathogens operate, how they interact with each other, so knowing our enemy, but also how they interact with other microbes associated with the plants. So some of those being beneficial, so how we can embrace our friends to try and produce healthy plants and productive plants. So as a plant pathologist, my research, or the research of my group, fits into the international and national strategies on providing sustainable strategies to reduce threat and impact of plant diseases on our ecosystems. And this is both on our productive ecosystems, but also on our native ecosystems. And some recent FAO data has estimated that about 20 to 40% of global crop yields each year are reduced by the impact of pests and diseases. And I haven't even got weeds in there, so any weed ecologists there, sorry, I haven't mentioned weeds. Increasingly, agriculture is focused on trying to reduce the pesticides that we're using to control these. And currently we're using about 3.5 million tonnes of agrochemicals just to control our fungal pathogens in agriculture. And increasingly these are becoming less effective. So I think I'm going to go to this one. So some recent data from Sarah Gur's group in the UK has shown that increasingly we need more pesticides to actually control our pathogens. So for Cymoseptoria triticide, the wheat pathogen, we need increasing levels. And that's because some of our pests and diseases are becoming resistant to these pesticides. So with the research of our group and other plant pathologists worldwide, and as one of my colleagues used to say, plant pathologists feed the world, and that's what we're trying to do. So trying to increase our productivity with minimizing our environmental impact, or trying to minimize the pesticides we're using and our environmental footprint. So the work that we do here at Lincoln is trying to reduce the pesticide usage by understanding the pathosystems, understanding how these pathogens operate, and then trying to then develop some integrated pest management strategies for these pests and pathogens. In the 20 years I've been at Lincoln, and I know it's quite hard to think that I've been here 20 years, it feels like yesterday, but I've worked on a range of different pathosystems, from forage crops, so dry rot of swede, downy mildew of peas and broadacre crops of peas and wheat. I've also worked on Sclerotinia, and this disease has followed me through my whole career. So I started here in Lincoln as a postdoc with Alison Stewart's group, working on biocontrol of Sclerotinia, went back to the UK, worked on Sclerotinia, and recently I've had a student, Maddie, who's just finished working on Sclerotinia, trying to develop an integrated control strategy for this pathogen. As well as these sort of broadacre crops, I've also worked on horticultural crops, so blueberry, blackcurrant, kiwi fruit, and also apple. And we've got one of my other students over there, Miriam, who's just about to submit her thesis in the next couple of weeks, looking at Phytophthora crown rot of apple, looking at the pathogens involved, looking at the resistant and tolerant cultivars, and also some biological amendments. But most of the diseases that I've talked about here are in that classical sort of single pathogen, single disease model. But increasingly, as plant pathologists here, know that we don't just get one disease, one pathogen, one disease on a plant, we get numerous pathogens. So we're going away from that sort of one pathogen, one disease model to looking more at these sort of complexes. And this is really where the work on grapevine trunk disease, which is where I really want to concentrate most of this talk on. So grapevine trunk diseases. So this is a term that encompasses a wide range of woody trunk diseases. So things like Utica dieback, Botrosphaera dieback, Esker, which luckily we don't have here currently in New Zealand, but also things like Petri disease and Blackfoot disease. So it's a complex of pathogens that often interact and affect these vines. But as well as being disease complexes, the pathogens involved are also complexes. There's many, many pathogens within each one of these diseases that interact and affect these vines. Two that I've worked on mostly here at Lincoln is Botrosphaera dieback and Blackfoot disease. So Botrosphaera dieback, you can see here, it causes, as the name suggests, this sort of dieback trunk decline. And if you cut into the cankers that develop, you get these very typical wedge-shaped lesions. These generally are old vine disease, or these symptoms develop and spread quite slowly in the vineyard. And you generally see these diseases in vineyards over 8 years old. And then some of the work we've done here at Lincoln has shown there's a wide range of pathogens involved from different genera, so number of species within Euphysicockum, Diplodia, and Botrosphaera species. So again, they're a complex within a complex. The other disease is Blackfoot disease. And as the name sort of suggests, you get this a below ground rotting of the stem base and the roots. You get this reddish purpley rot. This is a soilborne pathogen, and these are widespread. So they can be found as saprophytes in soil, on dead decaying material. They're also root colonizers, so there's some evidence that they can be endophytic, just colonizing those roots, and it's when the plant is stressed they become pathogens, or they can be pathogens. And again, the work here at Lincoln has shown that there's a number of different species within the genera Dactylonectria and Ilionectria that are responsible for these diseases, and often they coexist. So if I just look at Botchosphera dieback, so this is the group, as you can see, of a number of students here. So Botchosphera dieback, these are latent pathogens. So as we said, that they often we don't see them in the vineyard until the vineyard is about 8 years old. So these are latent. But this is the work of Reggie at the end here, who showed that many of the nursery propagating material that's been sent out to the vineyards are infected with these pathogens. So if you look at the figure here, these are grade 1 plants, the ones in white. They are the best, or the high quality vineyard material, and they're already infected with these pathogens. So they're being sent out from the vineyard into the vineyard, introducing that disease. These pathogens have a very wide host range, both in the nursery, where there's lots of common hosts to these pathogens, but also in the vineyard as well. So most woody plants can be infected with these pathogens. There's a common environment of pathogens. These are wound pathogens, and the susceptibility of those wounds are susceptible all year round. So looking here, this is different seasons, they're equally susceptible. And also, more importantly, these wounds remain susceptible for about 40 days. So those wounds, after they've been created, will be susceptible for a large number of days after that wounding event. To add to this is that conidia are produced all year round. There's an inoculum to infect these wounds present all year round. And the conidia are formed, or they're oozed out from the conidia on this disease material after rain, and they can be trapped all year round. So we can see, this is Nicholas's work here, looking at how they're being trapped all year round, and after as little as 0.6 millimeters of rain. And then some of the work that Amna did show that these can be spread quite some distance from that infected material, so they can quickly spread through that vineyard. The other issue we've got is that the current control is quite limited. We're really looking at using pesticides and fungicides as wound protectants that only prevent infection. But we've also got the issue that there's conidia all year round, and the wounds are susceptible for long periods of time. So very difficult to control. And additionally, we can't eliminate existing infections. The other disease, Blackfoot disease, again, a suite of students, and it seems to be an all-female team for Blackfoot. I'm not sure why. This is a young vine disease. This affects vines in the nursery or in young vineyards. And again, the propagation material can be infected by these pathogens. So again, we can be introducing these into the vineyard. This is a soilborne disease. All the propagules they produce are infective, and we can see sort of here that all the chlamydospores, conidia, and mycelium, but the conidia seem to cause higher disease severity. Again, this is a wound pathogen. So in the nursery, there are lots of wounds being produced that these pathogens can infect. So here, sort of incomplete grafting, or roots that have dropped off from graftlings. But also in the vineyard, so wounds created by nematodes or insects feeding on that below ground can also be entry points for these pathogens. There's an indication for this pathogen that stress increases the susceptibility. Generally get greater disease in poorly drained soils. So we can see in clay loam here, we've got more disease than in the more sort of free draining sandy and silt loam. Water stress, so when we've got waterlogging, that increases the disease compared with 75% field capacity. There's also an indication that these can work together. So this is coming back into that disease complex. when Caroline was doing her survey, she found a lot of plants infected with Petri disease, another pathogen when we could find cylindrocarpon. So some work that Chantal did, where she looked at vines that were already infected with Petri disease and compared them with ones that weren't, and found that when they were challenged with cylindrocarpon, we got higher disease levels in those, showing that these they can be working together, maybe opening the door for other pathogens. So because the propagation material is such an importance for the vineyard of introducing these pathogens into these vineyards, both Reggie and Caroline did some work on looking at hot water treatment. Can we eradicate these diseases from these vines? So Caroline did the work on Blackfoot disease, and she used naturally infected rootstock material of 2 cultivars, 10114 and 5C. And she used a range of different hot water treatments, some of them what the nursery is already using, and then trying to see if we could actually reduce the temperatures as well. And what she found was that we could eliminate the pathogen by using 30 minutes at 48.5 or 30 minutes at 50 degrees. And this was lower than what the nurseries were using. But all of these temperatures, even lower temperatures, significantly reduced the amount of infection. And there was no impact on plant growth of these hot water treatments. Reggie did some work on Botryopsera dieback, where again she had some infected canes. But what she found was that Botryopseriaceae, Neophysicoccum luteum, and parvum were a lot more tolerant of hot water treatments. So we could eliminate Neophysicoccum luteum, but not parvum, from vines that were hot water treated for 30 or 60 minutes at 53 degrees, but 30 minutes at 50 degrees didn't work. However, we could cure the infection, but unfortunately our patient died. So, which really doesn't help with So what we found was once we planted those canes, we got no bud burst or root development using those hotter temperatures. So with Botryopsera, we can't use hot water treatment to control this disease. We also looked at some work on fungicide dips and other work to see whether we could get rid of any external contamination. And we had some carbendazim and tebuconazole could actually clean these plant material up. Now, what we were really interested in was that there was some anecdotal evidence from the nurseries that there was some other negative impacts, as well as killing your vines if you use too hot a treatment, on these hot water treated vines. So this is the work of Fernan, who was an MFAT student. And what he did was he looked to see whether if we just hot water treated the vines as we would for Blackfoot disease, then we challenged them with Botryopsera. Was it going to make them more susceptible to Botryopsera? So he used Pinot Noir and Sauvignon Blanc canes, hot water treated them, he then rooted them, and then after 8 months growth in the greenhouse, he then inoculated them with Neophysicoccum luteum or parvum. Unfortunately, after that 8 months, when they came out of dormancy the following spring, the Pinot Noir really didn't grow very well, so we couldn't use Pinot Noir. But what we found was with Sauvignon Blanc was that when we hot water treat at the higher temperatures, either 15 or 30 minutes at 50 degrees, we get much higher disease progression. So these vines seem to be more susceptible to the Neophysicoccum luteum of parvum. So just to have a look, because I am a microbiologist after all, we wanted to look to see if there was any effect on some of the endophytic microbes that colonize colonize these canes. So immediately after, or a couple of days after hot water treatment, Fernan also isolated endophytic bacteria. And what he found was that about 3% of those could inhibit the growth of luteum or parvum on agar and deal plates. What was interesting was we recovered no bacteria that can inhibit these two pathogens at the higher temperatures, indicating that maybe these hot water treatments are negatively impacting that part of the community, those community that could suppress these pathogens. We also did some community analysis. This is some work that Romy helped with, just looking at those communities. But we did see a difference, but they're a little bit hard to sort of decipher. So what we've got here, these are Pinot Noir canes here, got very similar microbial community, a very distinct microbial community for Pinot Noir. Apart from the hot water treatment, which is over here in that light blue. So there seems to be an effect of the hot water treatment on that bacterial community. With Sauvignon Blanc, we're having a much more diverse microbial community and a little bit harder to see, but maybe an indication that hot water treatment is affecting those microbial communities. But again, need to have a sort of to work on that a bit more. So this sort of leads really into that grapevine microbiome work that I sort of alluded to at the start. So plants live in association with diverse community of microorganisms, collectively called the microbiome. And these will affect the health and productivity of those plants. So in that microbiome, we've got pathogens, which interact with each other. So the pathobiome. And with grapevine trunk, we've got the Utavilata, the Neophysicoccum that's shown in that sort of that diagram there. But as well as that, we've also got the beneficial microbes within this community that can also suppress these pathogens. So we've got microbes like the ones that Fernan shown that can inhibit those pathogens. We've got ones that can antagonize, they can parasitize, such as microparasitic Pythium species. And we've also got some beneficial microbes that are symbiotic with the roots, so the AMF fungi. And it's really that sort of balance between the pathogens and also stress and the beneficial microbes in that microbiome, which really determines whether we get disease plants or whether we get healthy, resilient plants. And that's where Nuruddin's work came in. So Nuruddin has just finished his PhD, and he's been characterizing the grapevine trunk microbiome. So this is the fungal component of that microbiome. What he did was he compared organic and conventional vineyards, old and young within the conventional, and then in each one of those vineyards, he had symptomatic vines to grapevine trunk disease and asymptomatic vines, the ones that didn't have disease nearby, so these paired vines. He then sampled the woody trunk, so he peeled off the back and then sampled that woody trunk using just a drill. And then he used that wood material to culture the culturomes. These are the culturable microbes. And we can find in those, we can see some common pathogens, so Utavilata, Diplodia, Eupheo, etc., that are found in that culture collection. But as well as doing a culture collection, he also looked at the total total fungal community in those vines using high throughput sequencing with Illumina-MiSeq. So he compared those factors that we talked about earlier. So he looked at the age. Does the age of the vine change the microbial community? And what he found was, yes, as we've expected, you get more, especially Utavilata, in those old vines. So we've got a pathogen. We know this pathogen is associated with old vines. So we're getting, so it wasn't unexpected. Looking at the health status, so these are the asymptomatic versus the symptomatic. Again, as we expected, we're seeing Utavilata in those symptomatic vines. It's a pathogen. But also what we were also seeing was we're seeing some key microbes that are in higher abundance in the asymptomatic vines, tending to suggest that these may be associated with that without the lack of disease. So Epicoccums, Oribasidium, and Somatosporium. What was interesting was that we didn't find any major difference between management practice, between organic and conventional management, which may be thinking about where these microbes are residing. They're residing inside those vines. They may be protected somewhat from the activity of pesticides. We have got some systemic pesticides, but again, we're not finding much difference between the conventional and the organic. So based on some of these microbes that were, or these fungi that were coexisting. We wanted to see whether there was any interaction between, starting to look at that pathobiome. These were just simple co-inoculations, but Nuruddin did a whole range of different co-inoculations. But one of the ones I wanted to talk about was with Somatosporium vitis and a new species of Somatosporium. So there's been some debate in the literature about whether these are grapevine trunk pathogens or whether they are associated with just a normal healthy microbiome. So what we did, or what Nuruddin did, was to co-inoculate Neophysiococcum parvum, which is highly pathogenic. It's one of the most pathogenic of the Botrysphaeriaceae that cause Botrysphaera dieback, and also Diplodocus areata, which is quite a weak pathogen. And when inoculated either Somatosporium vitis or species nov on its own, we didn't get any lesions. So don't seem to be pathogens on their own. But interestingly, Diplodocus areata, either on its own or with the other two, also didn't produce any lesions, which seems to match what we found in other work in our group, that the New Zealand population of Diplodocus areata is not really highly pathogenic compared with, say, in Australia, where it's one of their most important pathogens. But interesting for Neophysiococcum luteum, this, as expected, produces nice long lesions. There's no synergistic effect of the co-inoculation, but both of these Somatosporium reduce the lesion length. So they seem to be impeding those pathogens, that pathogen from causing the disease symptoms. But the role of Somatosporium was still really unclear about what their role is in grapevine trunk disease. And I'll come back to that in a little bit later when I talk about some other work we've done. Looking at the below ground partners, so we've also looked at arbuscular mycorrhizae, fungi. So these are symbiotic with the plant roots. Grapevines are highly responsive to these below ground partners. These will colonize, they'll extend the colonization volume of explored soil by the plant with their hyphae going out into the soil. They improve water and nutrient uptake. They therefore improve shoot and root growth, and they can also enhance disease resistance. This symbiosis is really important for plants. It's so important that the plant actually contributes or allocates about 4 to 20% of the photosynthetic carbon to that fungal partner. So really important, they're investing a lot in these partners. However, if the cost of that carbon to the plant outweighs the benefit, you can get negative growth response. So there is a balance there. And this seems to vary with rootstock, but also with the AMF, of which AMF and rootstock partner. So this is the work of Romy that she did for her PhD, looking at the factors which affect these AMF communities. So Romy sampled what I call soil, but I think Romy has classified as being concrete because it was so hard to dig, in a range of different vineyards, isolated the roots, did pot cultures, but also did DNA extraction and AMF community analysis. And what she found was that the main driver of the AMF community was actually the rootstock cultivar. We can see this really clearly here in Brancott that each of the AMF communities associated with the rootstock are tightly clustered. We can also see that soil will influence that AMF community. So we can see that here where we've got 3 different soil in the Camshaw vineyard. So we've got C, they cluster together, apart from 3309s in soil type C. That seems to do a slightly weird thing. We're not quite sure why that one seems to group with the rootstocks in the other soil type. Interestingly, just looking at organic and conventional, there was no effect of vineyard management. So that didn't seem to affect the diversity of those AMF communities. But what does this mean for grapevine health? So this is some work that Carolyn did, where she looked at colonizing different rootstock with different AMF species. And this was in a pot trial. And then she challenged, or she planted them in Blackfoot infected soil. So this is, So this is without Blackfoot, and this is with Blackfoot. And there seems to be an interaction between the AMF and the rootstock with regards to the disease that we found. A-Lelis inoculation of 5C seemed to decrease the disease, similar with Vanilliformis mossae. When Schwartzman, we tend to get decrease in disease. However, interestingly, Vanilliformis mossae inoculation of Rhiparia glare seemed to increase disease. So there seemed to be this interaction between the rootstock and the AMF. And some other work that Carolyn did was she then pre-colonized plant material with a community, this time of AMF, and then she planted them in a nursery site with Blackfoot infection. This time we didn't see any disease, but what we did see was in the ones inoculated with AMF, a 214% increase in root dry weight. Now, when Carolyn brought this data to myself and to Hayley, that was the co-supervisor, we thought it was 21.4, but it was 214, so a massive increase in root dry weight. I'm going to skip that one because I think I'm going over. So sorry, Jane. What we then looked at was we wanted to now start looking at microbial communities. So in the field of biocontrol specialists get very, very excited when they see a healthy plants in otherwise devastated crops. So where there's high disease level and we see individual plants that are surviving, we get very, very excited. So historically, that would be a way that we could find some resistant cultivars within a crop. Now they're generally similar genotype or they're clones, especially in vineyards, and they're grown in a similar habitat. So it's probably not to do with that. But what could be is a microbe or a microbial community that are protecting those plants from disease. So this is where the disease escape concept comes from, where we find healthy plants under high disease pressure. Similar genotype, in vineyards we get clones, and they're grown in a similar habitat, so they're grown in a block. So this is the work that Demola did, and he just defended his PhD on Monday, and he can talk to you on length on this work. So what we did to try and identify some of these vines that are escaping disease was to use some data from Dion Mundy from Plant and Food. So he had done 3 years of mapping vineyards, walking up and down the vines, mapping the disease level. So he produced these beautiful maps of the disease. We also used some anecdotal observations from viticultures to identify some vineyards, both in Hawke's Bay and Canterbury, where there was high disease and some disease escape potential. And then Demola went out and had a look at these vineyard blocks, and he assessed whether there was, in fact, lots of disease. He rated those vines on whether they were healthy, and also looked a little bit on the chlorophyll level as well. So based on that, he selected some plants. So like in the picture here, where we've got these plants here, this plant here is a potential disease escape, and surrounding it, we've got disease plants. And then using the same method that Nuruddin did, he sampled the wood of these and then looked at the total microbiome. So using high throughput sequencing with Illumina-MySeq. He looked at the fungal community, and this is just a summation because he's got lots and lots. This is me summing up lots of his data. So what we found was, as expected, we got disease in the disease. So we got Utaperlata in our diseased vines. But interestingly, we also got some higher prevalence of some other fungi in our disease escape. So we got Alternaria, Aurea basidium, and also Somatosporium, which comes back to Nuruddin's story. We also looked at the bacterial community, and what we found was that we could actually see some bacteria and higher prevalence in the disease escape. So Hymenobacter and also Pseudomonas. Well, when we looked at the pseudomonads in more detail, we could also see that there were variation within those. So this big genus pseudomonads, when we looked at the amplicon sequence variants, so these are sort of the individual different sequences and cluster them, we can see that there was differences. So in the diseased, we're seeing this one here, 43% of this particular ASV in the diseased, but only 0.01 in the disease escape. And similarly, we've got this here and this one here, which are mostly found in that disease escape. So the question is whether these differences are the cause of the disease escape, or is it the fact that we've got disease that's causing these differences? So could it be that we've got disease in these vines, which is causing these differences in, or are the disease escape actually being protected from disease? So the next step for Demola was to try and inoculate these plants. Can we mimic this disease escape? So he used this decision tree to select microbes to use as a synthetic community. So based on that, he selected 7 bacteria, some Pseudomonas isolates, an Aurea basidium, and a Somatosporium. Now we want to inoculate these plants. We want to get rid of the current microbiome. So we can't completely, but we reduce the microbiome using hot water treatment, not the 53 degrees that kills the plant, but a much lower level. And then we replace them with a selected community using a vacuum filtration method here. Canes were then rooted and then inoculated with the GTD pathogens, Utavilata and Neophysicoccum parvum. Sorry, luteum. Looking at the fungal communities, which were not here, we didn't really see a lot of establishment of the fungi. Some of the Aurea basidium established, but we couldn't detect the Somatosporium vitis. However, the bacterial synthetic community established really well and persisted for that 3 months. And we can see these here, yellow here, are the Pseudomonads here. What was really interesting was that we seemed to get better establishment when there was a fungus also inoculated, whether it was the fungal community or whether it was the pathogen here. So there seemed to be a synergistic effect there. We're not quite sure why that would be. Looking at the disease assessment, so looking at the disease, the colonization length, sorry, the lesion length, we can also see that this is the lesion length in the control. We're getting a reduction in both the fungus community, the fungus and the bacterial community. The bacterial, there's a few outliers that sort of affected those results. So we are sort of seeing some reduction in disease there. Again, I'm probably, sorry. So where do we go from now? So where do we go? So we envisage maybe a new product, a designed polar biomes, where we actually pair the plant genetics with the best microbial partners. So we could design a synthetic community of both above ground symbionts and below ground symbionts that we can inoculate these plants. But how could we do this? So we could mine some of our sequencing data. We've got a large amount of sequencing data. We could extract from our culture collections and maybe do a bit more sort of going back into those vines to actually find some of the other partners that we may not have. We could then design that synthetic community. We could test its efficacy, establish it, maybe not, maybe looking at different methods for establishing this committee, maybe tissue culture. And then we can then test their persistence. We only did it for 3 months. So we have got some vines left in the greenhouse that we might be doing some more on, and then test that magnitude of effect. And hopefully we can actually produce vines for the industry that are able to resist those diseases. So I just want to acknowledge my many colleagues. So Hayley and Marlene, who I've worked closely with on grapevine trunk diseases, Siona, who co-supervised a large number of students with me, Obviously Sandy, which we could not do any of this work without Sandy, that runs the group tightly, makes sure that we obey the rules and helps us out. But also some colleagues from Plant and Food, so Simon and Dion. The many students who, a lot of them are over there. So yeah, thanks to all of my students, and also the funders as well that have supported both the grapevine trunk disease work, but also the other work in this group. And thank you.
Superb on timing. It provides,
yeah, whiz through.
No, it provides an opportunity.
Those two slides then.
That's so fine. There is an opportunity now to ask questions of Eirian. Just commentary around to a presentation. If you just would note your name as you ask the question, please. Next.
David Jackson planted the first grapevines at Lincoln University, and one of the classes I looked at in the early 1970s. Would any of those vines be alive still?
I'm not sure. Romy, do you know if any? We've got the David Jackson vine, but I think they've been replanted, haven't they?
They've been pulled.
Yeah, so I don't think we've got any of. No, I don't think we've got any of those really old, sort of 50-odd-year-old vines.
So
unfortunately, probably some of the work that Marlene, Haley, and I did on spreading disease probably didn't help.
Maybe he should have gone to. David should have gone up to Ashley Dean where it was stony and there'd be less disease. Would that be fair?
Sorry,
there'd be less disease than the light stony soil of Ashley Dean.
Potentially for the Blackfoot disease. I'm just thinking, yeah, potentially Botrosphaeria if you didn't introduce it, because it depends on the problem we have here is we've got lots of different horticultural plants that all have Botrosphaeriaceae. So yeah, they can spread from one to the other. So maybe Ashley Dean would have been a good place to put the vineyard. I know they're looking at redeveloping the vineyard, so maybe that could be a plan, Romy.
Eirian, while some others are thinking questions, a couple of places you pointed to a comparison between organic and conventional.
Yeah.
What was your hypothesis that there would be a difference there, and were you surprised that there was no difference?
So we know that with foliar pathogens and foliar microbes, we do get a difference between conventional and organic. A lot of the chemicals we're using are going to be impacting because they're not just as active against the pathogens. I don't know what's happening in the vineyard, but I think that Romy has been doing some other work. I think there's a difference with some of the rootstocks. So some of the rootstocks seem to be better in the organic and seem to have a better community associated with them. So they might be, we could select some rootstocks for organic versus conventional, so that might come out of there. But I think when we're looking at pesticides and soil, it's very hard to, you're not going to affect all of that soil microbiome with pesticides. Yeah, not sure that answers.
I see in your isolations, you did get a lot of Pheromonia and Utipa, whichever you like to call it. Do you think these are emerging diseases, or are they just in the background being? So,
I think Utipa is definitely a pathogen, and it we seem to find it in some regions more than others. I can't remember, was it we found it more in Hawke's Bay? Yeah. Pheomola more in Hawke's Bay, and Utipa was more in Waipara. Pheomoniella is a really interesting one because Pheomoniella is associated with Esker. We don't have Esker because we don't have the Basidium mycetes here in New Zealand. But we find Pheomoniella everywhere. So whether it's a real pathogen or whether it's something that helps other pathogens get in, whether it's a sort of a gateway pathogen, we don't know. But we don't seem to see it only in disease. We seem to find it associated with everything else. And if we did get those Basidium mycetes, then it might be a problem. But yeah, And in the data, we weren't seeing it just in the, like Utapa, we see it in the diseased. We don't see it in the sort of the asymptomatic failure. We're getting it everywhere.
Could I ask another question?
Yeah.
Please.
Botryospharia, all the Botryosphaeriaceae.
Yeah.
They seem to be in so many woody plants. I mean, right through from the one I was quite interested in, Willemia pie. I've actually found it in that from on Australia. Now, it seems to be there all the time, but it seems to, you call it a latent disease. I would call it a latent degrader, because as soon as in nature, this is my theory anyhow, that in nature, when the plant comes under stress, that fungus, which is latent in the and almost endophytic in the wood, will be activated, and because the it's anything that's resisting that growth is taken that away, and it starts to break down the plant in a natural way. So always it's like almost like a nature's way of breaking it down, but it's already on board. You know what I mean? And that's what I'm saying, because I was doing lots and lots and lots of work on rootstocks, and both the rootstock and the cymes, and almost all the rootstock stuff, they had Botryospharia, and that was taken straight from the mother plant. It had very little chance of actually getting infected. And so my theory was it was there all the time. It's in the mother plant.
It's definitely a stress, like stress does trigger.
And stress comes
some of the work that I didn't talk about that Nuruddin has been doing is looking at that stress, looking at carbohydrate stress, and we've also looked at other stresses. And yes, we can get that. It's that switch from being a latent endophytic to being that pathogen. And yes, I agree with you. There's definitely a stress effect there, but what vines are under stress all the time in the vineyard. What is that sort of biotic or abiotic stress, and how does that switch over? And we also did some work on some natives, and we could find the Botryosphaeriaceae in things like Manuka. It's found there, but it doesn't cause disease. We don't see any disease. So we've also got whether some of our cultivated plants have been cultivated so much that they've lost some of their microbes that help to stop these pathogens expressing disease. If you're thinking of grapevines, they've been tissue cultured, they've been hot water treated, they've been all sorts of things, and maybe we're just losing some of that nice microbiome, that beneficial microbiome that suppresses those pathogens in that sort of domestication that we've done.
Interesting.
Other questions, Adrian?
Eirian, you've worked with a lot of postgrad students over the years to build this great research story. I just wondered if you had any insights of how you managed to keep everybody on task and you on task. Or some insights into what you might have done differently.
Be on task, that's a thing.
Well, put it this way, did you have a plan? Did you sit there and think, over the next 6-7 years, I'd like to go in this direction, or did it just organically grow, or how did you do it?
The problem I have is I didn't have a plan. I like students, I like plant pathogens, and if somebody gives me money, I'll work on it. So that's that wasn't my plan. It wasn't a plan, but I've always been interested in biocontrol and in soil and root diseases. So anything that but I'm not really.
Organically grew.
Oh, just organically grew. Yeah, you're about the same, Adrian. How many different? At least I've only worked on 3 kingdoms. So I don't know how many kingdoms you've worked on. Yeah.
Jim Morton, how much, when we look at a plant, how much of it is actually fungus?
I always wonder whether we always think of it as being the plant selecting the microbes, but is it the microbes selecting the plant and making the plant do what the microbes want them to do? There's always that, that the plant is selecting the AMF, or is the AMF selecting the plant? I'm not sure. Is the biomass? I don't know how much biomass would be. There's an interesting, I'll have to go and search and see if anybody's looked at the biomass.
Okay, I think there's 2 more questions down at the end. Too.
For the escaped plants, how much of a difference do you see between what you are able to sequence in terms of the microbial community versus what you can actually culture?
Quite a bit. Yeah, so there were some key ones that we couldn't put in a synthetic community because we just didn't isolate them.
Yeah.
We did try a whole range of different agars, but we are selecting the ones that are easy to culture, that like those sort of high nutrient conditions. And the problem with Botrosphaera that we've got is it's really fast growing, so it tends to, and the Utabe, it tends to outgrow everything else. So it's always a limitation of culturing, but the problem is if we don't culture, we can't actually do the other bits. We need to try and do both. And we can also, with those cultured, we can also sequence more genes and actually work out what we've actually got as well. But yeah, it's a good question. There's quite a lot. I think the estimate is about 90% of soil fungi can't be cultured, or soil microbes can't be cultured. So yeah, it'll be the same in the plant. We're probably missing. And there's a whole load in all of those microbiomes. You'll see the undefined and the unclassified. So there's a big group there that probably new to science that we don't even know what they are. Yeah. So a postdoc, Alana.
Do you have a question, Chad?
Just very quick one, Eirian. You ended this with the potential for novel approaches to intervention. Obviously, you're looking at a holobiome, which is very much phenomics based. What lessons can you drag from medicine into your approach?
Well, yeah, so know a lot about the medical side, but so I think we need to, at the moment what we're doing is looking at sort of who's there. We need to look at what they're doing. So looking more at that sort of transcriptomics to actually see what are these things doing in here, because just because they're there doesn't mean they're doing anything, especially in soil. So that would be one area. We have just put in an MV with Plants and Food to actually try and extend this work, and in that there would be some more of that sort of RNA work to try and find out what they're doing and seeing what effects they're having in the plant. So what pathways, what gene expression, etc., that they are doing in there. Yeah, so does that answer?
It does. Yeah, thank you.
Thank you. I think full time at that point. And, we have a small presentation for you. Included in there is the Lincoln University blanket, and by the look of outside, the change that is happening, temperature is probably applicable. There's a blanket and some other goodies in there. So thank you very much.
I needed a picture.
Please join us out in the Waimane for some drinks. Thank you very much.
Yes.